we're gonna talk about transcription. So first of all, what's the meaning of transcription? Let's remember together what the meaning of replication. Replication was formation of copies of DNA by using one strand of DNA as a template for the formation of the other. And replication was a semi-conservative process. Replication for the formation of a double helix. One strand is original and the other strand is new. Let's know what the meaning of transcription. Transcription is the process of conversion of a gene into a single strand of RNA. It's our first time to talk about RNA, and we said single strand. When we were talking about replication, we said double strand. We didn't say single strand. And we said DNA, but here we're talking about a gene. What's a gene? Let's remember together. A gene is a certain sequence in the DNA, but it's a piece of DNA that codes for a certain protein that carries instructions for a certain trait. Let's know more about transcription. Before talking about transcription, we should know what is the central dogma of molecular biology. What is the basic concept in molecular biology? When I say molecular biology, I'm talking about molecules. I'm talking about biological molecules like the DNA. What the general concept, the central dogma? The central dogma said for the formation of any protein like the protein of our hair color, protein of our eye color, protein for the formation of a certain enzyme. So the formation of many protein starts from the DNA. A certain gene in the DNA carries instructions for formation of this protein. But what are the steps? What's the central dogma? The central dogma says that the information starts from the DNA till the formation of RNA, after that the formation of the protein. So from DNA to RNA and from RNA to protein. If I said from DNA to protein direct, is it correct? No, it's not correct. The body or the cell should form first RNA, because the RNA is the molecule that can be translated into protein. So DNA to RNA to protein. This is the central dogma of molecular biology, or the basic concept of molecular biology, the direction of the information flow from DNA to RNA to protein. What's the name of the process of the formation of RNA from DNA? This process is transcription. Does it mean that there is another process? Yes. The formation of protein from RNA, this is translation. So the central dogma of molecular biology includes, number one, replication to get copies of DNA. After that, transcription to get RNA. Protein to get translation to get protein from the RNA. So, as you see here, the central dogma includes three processes: replication, transcription, translation. Where are those processes here in this figure? Replication here to form copies of the gene that the cell wants. After that, this copy will be transcribed into RNA, and after that the RNA will be translated to protein. Transcription, and here translation. Now we said RNA, DNA, are they same or different? First of all, we should know that both of them are nucleic acids, but they have some differences. Okay, so RNA differs from DNA in 3 major ways. What are those 3 major ways? Number 1, RNA has a sugar that's called ribose sugar. Do you remember the sugar for the DNA? Deoxyribose. That's why it's deoxyribose. But RNA are ribose sugar. Deoxyribose and ribose. Then, if you remember the 4 nitrogenous bases in DNA: adenine, thymine, cytosine, guanine. For RNA, they are different only in having uracil. So RNA has uracil instead of thymine. So we will not find in the sequence, nucleotide sequence of RNA, we will not find thymine. Instead of thymine, uracil. So what pairs with adenine in RNA? If there is A in the DNA, the RNA will form U instead of T. The RNA is a single strand. A single strand. Do you remember what was the DNA? The DNA, double helix, double stranded structure, while RNA single-stranded structure. Those are the 3 differences. This U means uracil. This is uracil. So instead of a thymine, there is uracil. Let's look again at this figure. The DNA versus RNA. Here is the structure of the RNA, single-stranded. The DNA, double-stranded. Deoxyribose sugar, there is no oxygen here. That's why it's called deoxy. In RNA, ribose sugar, there is oxygen here. Ribose sugar. The 4 bases in DNA, T, C, A, G. The 4 bases in RNA, U, uracil instead of thymine. Uracil, cytosine, adenine, guanine. This regarding to the difference between RNA and DNA. Transcription, as I told you, it's the process of formation of RNA by using DNA as a template. Transcription copies DNA to make a strand of RNA. What the name of the enzymes in transcription? The name of the enzyme is